Great. Well, thanks very much, Anne, for introducing me, and thanks very much for everyone and for logging on and listening. I think it's really a great opportunity to be able to speak at a conference like this. I think a lot of us in this current period are really quite starved for interaction with other scientists. So it's really great to see that this conference is going ahead, and it sounds like it's going great at the moment so far. So I just wanted to talk today mostly about comparing physiological patterns to patterns that we see in the wild. But before I get into talking about my research and some of these examples, I wanted to give a bit of an overview of how I got to my position. So I'm currently an assistant professor at Trinity College, Dublin in Ireland. And originally I'm from South Australia, and the environment that I grew up when I was a kid, I was fully immersed in the aquatic environment, and I very regularly encountered incredible aquatic animals. And I think because of this early exposure to these kind of environments and these kind of animals, it really informed my interest in the kind of work that I wanted to choose as a career. So to start with, I did a Bachelor of Environmental Science at the University of Adelaide. This was a 4-year degree with honours, and I think at the early part of my career, when I was trying to decide on what kind of education I wanted, I had a bit of an idea that I wanted to study environmental issues and animals, and I had an interest in marine animals, but I really wasn't sure early on what sort of career options would be most suitable for me. So I chose quite a broad undergraduate degree. And it wasn't really until I think the final year of that degree, that undergraduate degree, that I started to realise that marine zoology was something that really interested me. And so I chose to do a PhD in that field towards the final end of my undergraduate degree. And I really enjoyed it. I had an amazing time doing that project. And so soon after that, I was lucky to get a postdoctoral research fellowship over in Sydney. So that was I moved to a different state for that, and did a 3-year project looking at the general ecology of a range of different fish species. I then took a postdoctoral fellowship up in Japan. So I moved to Japan and did a 2-year project there, working at the National Institute of Polar Research. And again, I started, so I was mostly focusing on marine animal ecology and ecophysiology. And I started focusing more and more on large marine animal behaviour and ecology for that project. As that project wound up, I started trying to decide what I wanted to do next and where I wanted to move next. And I was lucky enough to get a postdoc after that, which was based in London in the UK. So I moved over to London and did a 2-year postdoc at the University of Roehampton. Again, I focused mostly on marine ecophysiology of animals. And while that was happening, I started looking for faculty positions. So I think, like a lot of people, I was really enjoying my postdoctoral research, but I'd done it for almost 7 years at that point. And I was trying to decide what I wanted to do next. And I was applying for several faculty positions in various places. And I was lucky enough to get offered one at Trinity College, Dublin in Ireland about 18 months ago. And so I guess my path really took me from when I did my undergrad. I then did a PhD in this field that I'm currently working in. But then it took me three separate postdocs in 3 different countries before I could get a faculty position in then a 4th country. So, I just wanted to highlight, I think, that particularly these days, if looking for faculty research positions is something that you're interested in, it's becoming more and more important, I think, that you gain international experience. So you gain research experience, or at least 3rd level education experience in various places to gain some sort of level of exposure to the ways that research is done and science is taught in different places around the world. I think that's important, and I think it's also necessary these days. I also wanted to just highlight a couple of things that I think are important if you're trying to look for faculty positions in science. I think my personal experience and my opinion is that there are several things that young researchers should be trying to focus on while they're looking to build competitiveness for these kind of roles, these kind of positions. And I think it's not just about getting great research papers and a lot of them. I think you also need to be focusing on various other aspects of your CV. So things like showing an ability to be able to attract research funding, generating industry links, I think, and having impact beyond academic impact, I think is important. And as I mentioned, having some illustration of your ability to move around and be a dynamic, type of researcher that can start new research programs from scratch, I think are all important things to be thinking about when you're a young researcher. That's my personal experience. And I'd also add that one of the big reasons I moved to Ireland is because my wife's Irish. So part of why I wanted to move here was for personal reasons as well. And I think that's often lost when early career researchers are thinking about, future careers. I think it's important to realise that, balancing work and life is really important. And I wanted to just point out that for me, that was a big part of what went into me coming to this part of the world. It's not always about work. So my research in general, I think, is mostly, I do quite a range of different things, but I think most of what I do these days is focused on the ecophysiology of marine animals. And I mostly study animals in their natural environment. So a lot of what I do for my research these days is out on the ocean, out on the water, where I study the behaviour and the ecology and the physiological status of wild animals in their natural environment. And something that keeps coming up, I think, a general theme that keeps coming up in my research is how the different ways in which environmental variation influence the behaviour and also the distribution of animals in space and in time. And a key element of this general question, I think, is how physiology mediates these relationships that we see in wild animals and their distributions. And I think that's why I was excited to be able to get the opportunity to talk to this audience for this conference, because I'm speaking to the next generation of outstanding young physiologists. So I thought it's an interesting point for me to emphasize. And I would say that in terms of different levels of biological organization, my perception is that there's often these disciplines of the physiology of individual organisms and population level dynamics and macroecological patterns that we see in nature are quite often, I think, studied in isolation. A lot of people do link physiological and macroecological or population level research together, but I think quite often these fields are studied in isolation. And I think part of that is because there's still a lot of uncertainty really about how well, if we study the individual physiology of organisms, how well we can use that information to then understand and make predictions about larger scale patterns that we see in nature at higher levels of biological organisation. I think most people would appreciate that these linkages do exist, and they do exist very strongly and in various complex ways. But I think there's still a large question around how well and how directly patterns in individual physiological levels of organisms can be used to then make inferences about these broader scale patterns that we see in nature. But of course, there are some really great examples of how this does provide a lot of really useful information for us. And I think that one of the classic ones that I think about is the metabolic theory of ecology, where it's perhaps still considered controversial in some ways, but nevertheless, it's been a hugely influential example of how exploring patterns, in particular scaling patterns at the individual level and even at the cellular level, can be used to help understand and certainly make predictions about some much higher level ecological and ecosystem scale processes. And the way I often think about, or I use this metabolic theory type example because it invokes not just individual level processes related to body mass and how body mass scales with metabolic rate, but also temperature. And I have in my research, increasingly, I have a focus on temperature. And I think the MTE is a really good example of how the temperature dependence of physiological processes can be used to explore mechanisms that drive broader scale ecological patterns. Now, I'm mostly a marine biologist, so I study fishes quite a lot. And I would say that research, there's an awful lot of amazing research going on at the moment with fish and with thermal physiology. So it's quite, for decades in fact, there's been a lot of research effort put into studying the physiology and physiological processes of individual fishes in controlled environmental settings. And we've learned so much from these kind of approaches. But I think it's still quite, I think it's fair to say that it's still quite controversial to take the various physiological understanding that we've developed in laboratory settings and scale those up to understand what happens in terms of how various environmental parameters structure population. A classic example of controversy, idea about how oxygen limitation regulates the distribution, I think is also mirrored in that there's an awful lot of fish researchers that study how temperature affects the ecology of wild fishes. So there's an awful lot of research going on where people head out and study patterns in terms of how temperature affects the ecology of fish in wild settings, in natural settings, but trying to identify and interpret physiological mechanisms that lead to those patterns in those ecological patterns are also quite difficult and also quite controversial. So I think scaling from physiological processes up to populations in the wild, and also scaling from population level wild processes back to physiological mechanisms can be quite difficult. And I think a big part of that difficulty comes from this uncertainty that we have about how well patterns that we find in terms of the individual physiology of individual animals translate up to these higher level ecological and macroecological patterns. So I want to just present now an example of research that I'm doing at the moment that tries to explore these relationships between individual physiology and population ecological level processes. And to do that, I want to just introduce this concept of heating tolerance. So we know that for most ectothermic animals, as temperature increases, performance, which can represent a range of physiological processes, growth rates, population growth rates, any type of performance metric that we can really come up with, we tend to see a curve that's shaped something like this, where performance increases to some high level and then decreases as temperature increases further. And so what I think of this, this decline, this rate of decline above some optimum level of performance as heating tolerance, because it represents how much additional heating beyond some optimum level that an organism or a population can tolerate. And so my overarching question for this study that I'm going to introduce was how does this ability for fishes to tolerate additional heating, how does this proxy vary for individual fishes in laboratory settings? So physiological processes of individuals, how does that compare to patterns that we see in terms of fish populations and the abundance of wild fishes living in their natural habitat? So to explore this general question, I teamed up with some colleagues from all over the world, actually, in particular, Simon Morley from Bass, and then also Amanda Bates, who's now at Memorial over in Canada. And I also got, so Simon and Amanda compiled hundreds and hundreds of measurements of physiological heating tolerance of individual fishes in in laboratory settings. And then I also got hold of a huge data set that was published by Connor Waldock last year, where they compiled a huge data set on how temperature influences abundance, so natural abundance of a load of different fish species, about 700, all across reefs around the world. And my general question was to compare how the heating tolerance of individual fish in laboratory settings compared to the heating tolerance of natural populations. So for this physiological heating tolerance, I was interested in looking at how much heating, how much acute heating individuals do in a laboratory before they reach some critical limit. So these critical limits are typically where the fish, so temperatures increased acutely, and they get to a point where some critical physiological function is lost. So this amount of heating, this difference in temperature, I was calling the heating tolerance of individual physiology. In terms of the analogy, the closest analogy I could think of in terms of abundance of natural populations was abundance of what we were able to compute was the difference between temperature where abundance was highest for each species in the wild, down to the highest temperature where that species is no longer found in the wild, so that species. So the difference between the temperature limit is what we call the, and I'm just going to important. So the first is that these have negative slopes in an individual fish in a laboratory. This is this top panel. As an individual fish are acclimated to higher temperatures, those fish tolerate a smaller amount of additional heating in the laboratory. So heating tolerance of individuals tends to decrease as fish are acclimated to higher temperatures. We see the same negative relationship for wild fish. So as natural fish populations in the wild, as those species have a have a high abundance at a higher temperature. So in other words, as their T-opt in the wild is higher, those populations tolerate less additional heating. So a fish that has a very high optimum temperature is only found at a small range of additional temperatures in the wild before their range limit is reached. So that's finding number one. The second one is that the heating tolerance of individuals in the laboratory and of populations in the wild decline at basically the same rate. So the overall slope of these two relationships is about 0.6. So in other words, every 1 degree increased acclimation temperature for fish physiology, individuals in the lab, and abundance, so as T-opt is at a high temperature, every 1 degree increase results in about a 0.6 degrees Celsius decline in heating tolerance. So very similar rates. And then third point, which is unexpected to me, and I'm still trying to understand why this happens. The third point is that at any temperature than temperate fish species, which are these brown, yellow, mustard colour, and any temperature either in the lab or the wild, a slightly higher heating tolerance than a temperate species. But I think that's address this general question of whether looking at different species in laboratory settings translate into terms of ecological patterns, so broader scale presented today. Yes, we do. We see not just a shared scaling of heating tolerance, so we don't just see that the slope of those relationships is the same across physiological and wild abundance data sets, but we also see this similar tropical temperate difference where tropical species tend to have a slightly higher intercept than temperate species. So we see those several different points of evidence that show we have these some similar scaling across those two data sets. And I think it's important and it's potentially useful to find these similarities because, to me at least, it's indicative of some sort of mechanistic link between physiological processes that happen in individual animals and population level and ecosystem level processes. In our case, it was looking at how abundance of animals changes in the wild. And I think if we can improve mechanistic understanding of how these ecological type patterns come of these natural. And I think, so I'm basically out of time and I'm basically finished, but I think there are some really great examples of how comparing large physiological data sets to ecological data sets can provide a lot of important and useful information. And I think one of the ways, not just in terms of improving understanding of individual patterns and processes, specific ones, I think by bringing together physiological and macroecological data sets, it also might help to bridge discipline gaps and get people working in quite different fields to collaborate more and work closer together. And I think that can only be a positive thing in terms of progression of science. And this is not a controversial statement at all, because you know, more and more we are seeing examples of how people are bringing physiological understanding and physiological mechanism into ecological understanding. And I think things like mechanistic species distribution models are a really great example, a great newish example of how bringing those different disciplines together can be powerful. And that's really all I want to have to say. I think one of the overall points, general points I wanted to try to get across by bringing together these different data sets and some of these ideas and theories, that temperature in general, I think, you know, has a really strong regulating influence, not just on physiology, but also on the distribution of animals. And I think most of us will recognize that, but I think the data sets I presented today are kind of interesting in that they show that the way that some of these processes scale can be quite similar in terms of physiology of animals and their populations. And I'm out of time in terms of my talk, so I'll hopefully have a few minutes left for questions, but I definitely want to just thank loads of my collaborators that have helped me work in this space for the last few years. And this is, you can read them on the screen there. So yeah, so thanks for listening. So I'd love to take any questions if anyone has any.
Thank you, Niklas, for a very interesting presentation. I want to point out for those of you that are joining the next session that you should join that now so you don't miss anything. But we got a lot of interesting questions, so we will go on with this session, and so Niklas can try to answer those questions. The first question is from an anonymous attendee from Oxford. Do the prey fish slow with increasing temperature in proportion to predator fish, or does the lower body mass enable them to dissipate more heat? You should unmute yourself, Niklas.
Sorry. Could you ask the first half of that question again? Sorry, I missed the first half.
Do the prey fish slow with increasing temperature in proportion to predator fish?
Oh, okay, sure. So yeah, that's a great question. So we didn't look at that level of information in terms of trying to explain variation between species. What I would say that in terms of body mass, I wouldn't expect body mass to have made much of a difference, because certainly in terms of changes in heating rates, because most of the fish in both data sets were, I would say, less than a kilo or two, and probably within one order of magnitude body mass in general. So yeah, it's definitely an interesting question. I wouldn't expect that the different heating rates in terms of their acute heating protocols in the laboratory, that they would have a huge impact on how rapidly, you know, differential changes in body temperature within the lab, because most of the animals were relatively small. But yeah, that is definitely something. I mean, you see in those plots a huge amount of residual variation that's not explained by our main effects. And I think things like body size, things like lifestyle, activity level, whether they're predatory or herbivorous animals, probably would explain some more of that variation. Yeah.
Okay, thank you. And the next question is from Philemon Michelia from ABTU in Nigeria. Your study was based on behavioral responses to high temperature. Are there responses to decreasing temperatures?
Oh, great. Okay, sure. So these were all heating. Yeah. So these were all so they not so much behavioral responses, but physiological and distribution responses. But I was only really interested in in the heating side because of my interest in how future climate are likely to impact these animals. Also, it's pretty clear from looking at physiological literature in terms of how fish respond to temperature. Unfortunately, there is quite a skew in available data between people that measure heating, tolerance of heating, compared to those that measure tolerance of cooling. There are a lot of really great studies that do look at that, but there is overwhelmingly more data from people that have measured responses to increasing temperature. And I think it's understandable in a lot of ways, given that most of the future climate change predictions are associated with increasing temperatures, certainly variability, and as well as things like average temperatures. But I think the available data probably reflects that. And so that was another part of why we focused mostly on the heating side of things, is because we wanted the greatest amount of power to explore patterns, and that comes from having more available data.
Okay, thanks. And another question: how do you suggest that laboratory physiologists can get started with taking their studies to the wild?
Well, I mean, that's a great question. And it's hard. I mean, it depends a lot on specific research questions. But what I would say is that these days it's an exciting time to be. I'm not I don't call myself a physiologist really. I sort of at most dabble in physiological questions a little bit. But I think for proper physiologists, I would say it's an exciting time to be studying animals, because in the last few years, we're seeing a huge amount of very exciting sensors and electronic equipment that we can use to derive physiological measurements from animals that are in the wild. And I think, I mean, even Anne, before I started my talk, you were mentioning your biologging work on moose, and I think it's one way that I think people that have an interest in physiology can get relevance in natural processes in the wild, is to use these new types of devices that can measure a broad range of physiological parameters within animals. And I think that's certainly one quite direct way of taking physiological theory and applying it to behaviour or distribution information in wild animals. I certainly try to do that more and more myself, using things like speed sensors and body temperature sensors, accelerometers and things like that.
Okay, thanks. And a question from Monica Trondrud from Norwegian University of Life Sciences. Can fish display behavioral responses to high temperatures to avoid heat stress?
Yep, sure they can. Yeah, sure they can. I think in natural settings, there are opportunities for fish to seek cooler waters, particularly if they are in an area where there is significant water depth variation. We do see that. We do see evidence of behavioural thermoregulation, or we see behaviour that's consistent with behavioural thermoregulation in fish. But it is certainly clear that fishes, while there is capacity for behavioural thermoregulation, there is a lot less capacity than there is for terrestrial animals, because of the way that heat is lost and gained very rapidly from water. So the ability for fishes to bask, for example, seek shade and various other microclimates is much less powerful than is the case for terrestrial animals. And I think that's an important reason why we we're tending to see more and more evidence that aquatic animals, aquatic ectotherms, are responding, their distributions are responding more rapidly to temperature change, climate change, than terrestrial animals. And I think a lot of that comes from the fact that it's much harder for them to buffer temperature variation through things like behaviour and microhabitat selection. So that's a really good question.
Oh, thanks. A new question from Laura. Do rearing temperature during fish development change the heat tolerance when adults?
Okay, so that's a great question. That also goes into that category of variables that we haven't really explored yet in much detail. Most of the animals in that physiological data set are wild collected, so most of them are collected from a particular habitat in the wild, transferred to a laboratory and acclimated in the laboratory for days and weeks and months. So I suspect that the location of collection, and particularly latitude, probably would have some influence on subsequent heating tolerance of those fish. But we overwhelmingly see that the main variation in those data sets comes from the temperature that they're acclimated to immediately prior to the heat ramping experiment, where the acute heating happens. Yeah, if that answers that question, I'm not sure.
Thanks. And the last question before we need to quit this session. How can terrestrial and marine ecologists collaborate or help proper understanding forward to understand these larger questions, like how do you scale individual physiological processes to population level?
Yep, so that's a great question as well. I mean, I'd say, yeah, there, you know, that my impression that there is that there is inevitably a, you know, people that are interested in marine animals tend to focus their work mostly on marine animals, and people that historically work on terrestrial animals tend to remain interested in terrestrial animals. That's certainly the case for me. But I think a lot of the physiological concepts and theories that have been developed in recent decades traverse habitats. I mean, a lot of the predictions we'd make about how these animals might respond to future change will be different given physical differences in environments. So like water versus air is obviously a big one. But a lot of the most, I think, fundamental questions that people are applying physiological understanding to in terms of understanding ecological patterns are not really habitat specific. So some of the things that people are looking at these days in terms of how temperature affects body sizes of animals, or you know, how oxygen limitation might affect animals in different habitats to different altitudes or water depths. I mean, these kind of concepts traverse habitats, and I think, yeah, I think my impression is that more and more people are starting to ask questions across habitats. I feel like it's kind of happening organically as we start to isolate the questions that we think are most important for climate change. I'm not sure of more active mechanisms that we could try to develop to bring terrestrial and aquatic scientists together, but you certainly have an example today where your speakers across this week are from various habitats. And, you know, while I have a focus on, my main interest is marine animals, I'm certainly very interested to hear some of the talks from people that are presenting ideas using terrestrial animals. So I think that combination of habitats is happening.